Everyone, thank you very much for coming in-house, and we're also finally streaming this successfully on Teams. We'll have to trim about 10 minutes of Ben panicking while he fixes the technology from the video that we send out. Anyway, we're here to welcome Rudolf Майєр. He's a professor of integrative biodiversity discovery at our counterpart, the Natural History Museum in Berlin, and also a professor at Humboldt University. Before that, he was a professor at the National University in Singapore for about 20 years, you were saying? Almost. Yeah. And he's the current chairperson of the International Congress of Dipterists, Dipterology. He's a dipterist, but we won't hold that against him. I guess there's a few species in there. And also the editor-in-chief of Cladistics. And he's going to tell us all about accelerating biodiversity discovery through various means. And I'll hand over to you. Thank you.
Well, thank you very much for the introduction and also for organizing this event, which was a bit stressful for Ben because, yeah, the microphone wasn't working until like 2 minutes before it started. Yeah, so this I'm going to talk a little bit about accelerating biodiversity discovery in hyperdiverse arthropod clades with techniques that we have been developing over the last 5 to 10 years. After 250 years of taxonomy in museums, but also universities, but lately more in museums, 90% of all species remain undescribed. 95% of all described species are probably only identifiable by 2-3 people on the planet. And we know next to nothing about what these species are actually doing. Maybe for 5% or so, we have information on ecology, and that makes it very difficult for us to do biomonitoring. And it also makes it very difficult for us to understand whether a particular change is important or not from an environmental point of view. This is all old news. So what is new? And I think this is a major change, which is that it's not a topic that biologists care about or only biologists care about. It's now a topic that is important also for society at large. So if you look at the World Economic Forum, they're doing these surveys about global risks on an annual basis. They have been doing this for more than 10 years. And biodiversity loss is now considered, based on likelihood and impact, to be one of the most serious threats to the planetary health. If you go back 10 years ago, it was already on the radar, but it was not considered to be a major threat yet. So what does it mean? It means that we have to also reevaluate what kind of biodiversity research we are doing, because we're no longer doing it for biologists, but we're actually doing it for society. And that brings up the question, are we studying the right taxa? And there are many different ways of looking at this. One way to look at it is to look at biomass. And this is a biomass distribution of terrestrial animals. And what you see is that in terms of biomass, arthropods and annelids are really dominating, while other popular groups like mammals or birds are overall not contributing much to the biomass. So if you take the point of view of saving biodiversity based on quantitative arguments, you would have to refocus your emphasis with regard to taxa. What's the current situation? So we can go to the Global Biodiversity Information Facility to look at where we have the largest number of occurrence records. And all of this here is birds. So the Global Biodiversity Information Facility is actually a global bird information facility. The number of records for geese and ducks is roughly the number of records that are available for insects. And the job is, of course, even bigger than we thought, the job of describing diversity. Because if you again take the biomass perspective, we are only like dabbling in this tiny little corner here. These are the animals. Plants, fortunately, are fairly well known, or seed plants are fairly well known in terms of at least species diversity. But there's these giant chunks here that are having large numbers of species or species-like organisms that are also very poorly known. Are we paying attention to the right regions? So we can also go back to the GBIF, or we can first of all restate the obvious, which is that the species diversity is highest along along the equator. It doesn't matter whether we look at vascular plants or we look at amphibians. When we look at GBIF data, in this case only for insects, we see the opposite pattern, where most of the data is found in the north and in the deep south, with the center here being data deprived, despite the fact that it has the largest numbers of species. You also see a very strong west-east gradient here too. That's one that is very rarely discussed, but it is at least as pronounced as the one that is from north to the south. So that's pretty dangerous because biodiversity loss is starting to be a major issue globally, and we are having very little data for those areas that have the largest amount of biodiversity and arguably will be affected by biodiversity loss to a larger extent than the countries in the north. Does this data divide also exist for all kinds of data? So if you look at iNaturalist data as a way of looking at citizen science, we see that there is even more of a bias in citizen science towards the north. If you look at machine observations, which will be increasingly important for getting biodiversity information on a regular basis, we have an even more dramatic bias. So the techniques that are yielding the largest number of observations by time are particularly poorly established in those countries where we need to gather data. Is the situation improving? This is like the number of GBIF records over time for Europe and Central Asia. And this is for Africa. At first, it looks about the same, but the y-axis is dramatically different. So if we shrink the y-axis, then this is what it is. So with other words, the major gap between knowledge for the north and for the global south, although I personally think it's more like the global middle, the gap is getting bigger and bigger and bigger. So that means we have like the obligation here to really overcome this data bias. And for that, we need to have cost-effective data collection techniques. It can't be super expensive stuff. The most cost-effective method, arguably, is image data, because you can convert image data into biodiversity discovery information if you have the right kind of convolutional neural networks for doing species identifications. Acoustic data is also quite attractive in that sense. Sequence data much less so, because it's quite expensive to generate. The techniques have to be easy to learn and should require very little infrastructure, and training should then be made available to people from all over the world. So this is what we are trying to do in the Center for Integrated Biodiversity Discovery at the Naturkunde Museum in Berlin. We try to address this, which is a pile of insects coming from a Malay strap. The traditional way of dealing with that is to pick out maybe 10% of the specimens and then leave the rest and put it somewhere into storage or throw it away. We want to actually deal with the things that are down here and that are in storage by the billions. And the second part that we want to address here is really how do we actually get more information for countries that are deprived of biodiversity data. And that means we need to develop cheap ways of getting data. So what we need is really frugal techniques for dark invertebrate taxa. What are these dark taxa? Well, these are they're represented by large numbers of specimens. It's the kind of stuff that nobody wants to work on when they get a large sample. Large numbers of this species. They contribute to a large amount of functional diversity. It's usually not being treated because morphospecies sorting for 10,000 specimens at the same time that contain 500 species doesn't work. You can do this for smaller numbers of specimens, but once you have like a huge number of specimens and a huge number of species, and you try to sort into the different petri dishes, whoever has tried this before knows that it's extremely difficult, time-consuming, and it's not really a solution. The definition of dark taxa that we have recently proposed is more a definition of what is not a dark taxon, which is it's a clade where less than 10% of all species are described, and the estimated species diversity will exceed 1,000 species. And I would think that there is a fairly small number of taxa that fit that bill, but it's the taxa that are most neglected because of the lack of techniques for large numbers of specimens. Now, I would argue what we need is a dark taxon biology as a new field in biodiversity science. We cover everything else, but these like tiny little things that are so numerous, have so many species, those we are ignoring. How many of these specimens how many of these species exist, and how many specimens do get collected by, like, for example, Malay strap samples? So we're looking here at Sweden. These are like 36 Malay straps that were run for 2 weeks in the summer. On average, we obtained 1,000 specimens of phorids, which is one of those dark taxa, per trap. If you were to now run a thought experiment, if you were to run 10 traps in each of the 450,000 square kilometers in Sweden, you would get within 2 weeks 4.5 billion specimens. And no, we don't know anything about them. We don't really know what they're doing and how much they move around and what microbes they're spreading. Continue with this thought experiment, and you assume that only 1% of the flies are stupid enough to go into a Malay strap, then we would come to the conclusion that there is at any point in time 1 trillion phorid flies going around Sweden. And yeah, we don't really know what they're doing. We know for some species that they are pollinators. We know for other species that they're parasitoids. Some of them are even phytophagous. So they're doing things that are very likely to be relevant to the environment, but we have been neglecting them for hundreds of years. And they could be very useful for all kinds of purposes, among others also for monitoring climate change. This is like a Sweden garden zone map. It tells you what vegetable you can grow in what part of Sweden. So it's like a mixture of temperature, precipitation, and also the season length. And if you sequence 31,000 phorids, then they fall into about 400 species. Then you can actually use an NMDS plot to show how the communities are spread across Sweden. And what you find is that that is a very good representation of the Swedish garden map, which implies that you are here having a tool for monitoring climate change, because phorid species communities are responding to those factors that are also important for vegetable growth. How dark can dark taxa get? This is a particularly extreme case. Again, phorid flies. This Malay strap was run for 2 years in a Ugandan national park. 8,000 phorids were found in 8 weeks. They consisted of 650 species based on just looking at the barcodes. 250 years of taxonomy have only yielded 466 species for the entire Afrotropical region. We can again do like some crazy thought experiment here and assume that if every 100 square kilometers have 100 endemic species in Afrotropical region, then we would expect 200,000 species of phorids in the Afrotropical region only. Whole areas, whole habitats can be dark. So this is an experiment that we did in Singapore, where we worked on mangrove insects. Mangroves were considered poor in insect diversity because it's a saline environment, and the general knowledge or the general idea is that insects are not very good at dealing with saline environments. But when we started to do a survey in Singapore across many different habitats, and a large number of Malay strap samples were put up, and we sequenced about 60,000 specimens there, we find that mangroves here are contributing a very large number of species to the overall species diversity in Singapore. Now, a large number of species doesn't mean anything if these are just common species that are found everywhere. So we need to not only look at large numbers of species, but we also need to look at whether these species are unique to mangroves. And for that, we can again look at NMDS plots. And all these green points here are mangrove insects. And then these other points or the other color schemes are for other kinds of forests. And what you see is that the mangroves are having a very distinct and a rich fauna. And that's regardless of whether you remove singletons or you remove every species that is only known for less than 10 specimens. So it's a robust result. And mangroves are disappearing very fast. So we basically discovered here that there is a huge insect diversity in mangroves, and that it's an environment that we will not see much longer for many different reasons. So this is an old challenge here. This has a new significance, and now we need new tools. So how can we deal with dark taxa? And this is something that we have been doing over the last 5 years of developing new techniques, frugal techniques. And there's 2 strategies. One is innovation through subtraction, and the second one is innovation through technology. And I can only emphasize this first point over and over again. There is like a study showing that in science, we always go complicated instead of like trying to become easier, because oftentimes we can find easier solutions that are much more effective if we start thinking in that direction instead of always like looking for a more complicated solution. Of course, technology is nevertheless very important, and our favorite tool for sequencing, for example, are these MinION sequencers. So how do we do species discovery? So we have built something called a diversity scanner, which is a digitization robot. The sample goes here in the middle. Then the robot will find specimens that are far enough from each other to be treated independently. Then another camera comes and takes an image of that specimen. Then it sucks up that specimen and moves it into a microplate. Now we have a lot of images. So this can generate like 1,000 images a day for 1,000 different specimens. We don't know what species they are. So in order to get the species level information, we need to use another tool, which in this case is barcoding. And we're using MinION barcoding for that. Once we can then sort the images that were generated here, approximately to species level using the barcodes generated here, we're getting large image image sets that can be used to train convolutional neural networks. And that means we're going towards identifying species using machines. And we can skip this step as long as the convolutional neural networks are capable to identify the specimens based on images only. So the sequencing would be only an intermediate stage if we can get to that level. Just a bit more detail on those 3 steps. So this is like the diversity scanner. It images each specimen. It classifies it also at this point at family level based on CNNs. So it's not classifying it based on people looking at it. It measures the specimen and it prepares it for DNA sequencing. And here's the scanner in operation. So it does an overview of the sample. It then indicates where the specimens are and which specimens are far enough from each other to be pipetted. And here comes the pipette, picks it up, and brings it over into the well. Enlarge, this is the overview image, that's one camera. Now it has recognized that this specimen here can be processed. It takes another image, the pipette comes, picks it up, and moves it over into the microplate. These are the images that we're getting. So these are automatically acquired images, not images that are involving a human intervention. And these are, at this point, specimens that are only 3 millimeters and smaller, which are usually quite difficult to image, especially in alcohol. But with this system, we're getting quite satisfying results. Results that are good enough for doing some rough taxonomic work, but also results that are good enough for training convolutional neural networks. So it's also some biomass measurements being done. The length measurement is automatic. We are now in the process of also doing volumetric biomass measurements by measuring the size of the head, of the thorax, and the abdomen. For that, you don't want to have only one image. You need multiple images. And what is coming now is a new imaging device that takes images from multiple angles for 3D modeling. So this is the Bauplan. So the specimen is in the middle here in a capillary, and the camera goes all around it. And these are the images that you're getting. So you're getting dorsal, ventral, and lateral images for each specimen. We're also building new diversity scanners for larger specimens, and some of them will involve something like a diamond picker to pick up specimens that are too large for pipetting. We right now have already developed the imaging system, so eventually the imaging system and the robotic system will be combined. So we can right now doing it manually by putting an image specimen here, click a button, get the image, move the specimen into the microplate. In the future, that will be done with robots. Menopause sequencing is also something that is now being used in London very much for barcoding, from what I hear. We love it because it's fitting into your pocket, so you can do it anywhere you want. It also is allowing us to make this whole process of barcoding a democratic technique. So at this point, a lot of people will then say, like, oh, it's still too expensive, but sequencing for us is about 5 cents per specimen, so it's actually not that expensive. If you spend a lot of time looking at a specimen, that is actually more expensive. And of course, what we do is we use these barcodes to group specimens and images into putative species. That is about 80% accurate, 20% wrong, and we'll talk about the 20% a bit later. And then we can identify some of these putative species using blasts or sequencing identified museum specimens. This is a huge job for the next 20, 30 years to get all the types sequenced. And then, of course, we can also identify by describing the new species. So this is a part of, this is possible because we did some innovation through subtraction. We don't do DNA extraction, we do DNA leaching. We have simplified the amplicon production by doing a single PCR tagged amplicon reaction. We have simplified sequencing for Illumina and MinION. And we can now get an amplicon for less than 10 cents that we can then sequence for another 10 cents or 6 cents, depending on how you what kind of success rate you want to have. We can also sequence it for 2 cents if you do Illumina sequencing. And it's robust. So we have been doing this in undergraduate classes in Singapore for quite a while. And after one of the Saturday sessions where we do species discovery, we usually have 4,000 or 5,000 specimens sequenced. So the basic technique is tagged amplicon sequencing. So you have like 2 primers, you have tags, And then you can just multiply the different tags in order to get the number of specimens that can be pooled in one pool for sequencing. So if you have 105 forward primers, you have one plate of R primers, each of them individually tagged, then you can produce a total of 10,000 tag combinations, which you make each of the amplicons unique and assignable to a particular specimen. Of course, you also have to do innovation through technology, and I'll skip through most of this. Our choice now is to do ONT, doing Oxford Nanopore technology sequencing. We could do it with Illumina, but the largest Illumina machines, the NovaSeq, can actually allow you to put 400,000 amplicons onto one lane. Before you have 400,000 PCRs run, it'll take you a very long time. You have 400,000 specimens lying around. So although it's a cheaper way of doing things, it is also a totally non-operational way of upscaling. So that's why we're moving to ONT. Our experience with ONT is very good. So we can sequence about 10,000 barcodes in one min-ion flow cell if we assume 80% success rate. So why do I say 80% success rate? Because you have weak amplicons and you have strong amplicons. If you want to raise your success rate, you need to sequence deeper. Then you can only put fewer amplicons in your pool. And so you cannot really say sequencing costs X, because it depends entirely on what proportion of your pool you want to recover. If we go with 80% success rate and 10,000 barcodes per sequence per flow cell, the cost is about 6 cents. If you go for a ProMethIon flow cell, which has more pores and but doesn't cost that much more, you can go for 3 cents. Why do I like min-ion? Because it's incredibly flexible. The flow cells can be used multiple times. Every time you reuse a flow cell, the number of pores comes down, so the capacity comes down. So you can have like a bank of different flow cells in your freezer, in your fridge, where you have flow cells that are good for 2,000 specimens, flow cells that are good for 500 specimens, flow cells that are good for 10,000 specimens. You can also use Flongle for that. So Flongle is small device that costs $90 and can do about 250 barcodes, and 250 specimens can be barcoded with it. Outlook to me is very good. We have seen this evolving very, very rapidly. In a couple of years, you'll see 20,000 in one flow cell, and the quality of the reads will also dramatically improve. So if you run one of these experiments here, this is an experiment for the Afrotropical forest from Uganda. We had, in this case, Illumina data, so we could actually compare the quality of the barcodes from Illumina with the quality of the barcodes from ONT. If you see a solid green, it means they're identical. If you see a lighter green, it means that the ONT barcode has an ambiguous base, so an N, not a mistake, but an ambiguous base. And only those cases where you see red here, there was a disagreement between the barcode obtained with ONT and the barcode obtained with another technique. This is here by number of hours running the flow cell. And you see that most of it is actually obtained within the first 10 hours. So you don't really have to run that very long for a pool of about 3,500 specimens here. This is the coverage. So if you put on the x-axis coverage, we'll see that you get pretty good barcodes at 50x coverage, which also allows you to estimate how many raw reads you should be collecting if you have a pool with a certain number of amplicons. And this is like shown on this somewhat complicated graph, but it essentially tells you if you want a 50% success rate in your barcode experiment, and you want to sequence 250 barcodes, then you need only around 1,000 to 3,000 reads, raw reads, before demultiplexing. So it gives you like a chart for planning your experiment so that you are getting the right number of raw reads from the min-ion flow cell, because min-ion flow cells can be stopped at any point in time. So you can deliberately say, okay, 5 hours is enough for this experiment, and that gives you a larger number of open pores if you want to reuse that particular flow cell. Other ways of like improving the throughput is working with things like these next-gen PCR machines that can do 30 cycles in as little as 2 minutes by having 3 different plates that are at the optimized temperature, and by flattening the microplates, which allows you to have immediate transfer of temperature. And then things like dispensers that allow you to not only save in consumables, but also very efficiently dispensing master mix and anything that has to be dispensed from one container to many, many wells. Output is something like this, which is each of these are one specimen, each of the lines. So that's specimen ID, date, locality, and information. And then we have information on how distinct different species are. We express them here as percentages. So this is 6%, 0.6% away from this other specimen here. And then you can actually make a movie and look at how much work you have done. So this is like a movie for 350,000 specimens. And every time a line crosses my laser pointer here, a new species comes through. And there's about 9,000 species here. The movie is 45 minutes. So if you want to fall asleep, this is like the way to go. But you also see that it gives you information on abundance. It gives you information on how large is the haplotype diversity in species. So it gives you all kinds of information. And yeah, this information can be obtained rather rapidly now. Last but not least, so we used AI for doing the species identification. And what's quite interesting is that you can use these heat maps to find out what part of the body has been used for identifying that particular taxon to the class that was tried, that the CNN was trained for. And this is something that we will eventually want to explore for getting species diagnosis, or at least to get some hint to what are the diagnostic differences between close related species. Because that can also be automatically generated by algorithms. At this point, we are doing identification only to the most common groups in malaise traps. Most of these groups are family level groups. These are the success rates. So essentially, when the diversity scanner comes and takes the first image, within 2 seconds, it knows what family it is. And then you can potentially also move, let's say, only the specimens for one family into one microplate and specimens for another family into another microplate. The CNNs for common taxa evolve faster because you obtain more imaging, more images for the common taxa first. The diversity scanner can also be only moving specimens for one type of insect, which allows you to subsample a very large sample. So you have too many chironomids in your sample, you could say, just give me like 3 plates of chironomids, not 100 plates. So that's basically the outline. So we want to image, we want to know what species are represented. And then we can use this for the future, because in the future we want to do much more image-based identification and not sequence-based identification. So why barcode specimens and not entire samples? This is ultimately about like meta-barcoding. Why don't I do meta-barcoding? And there's really 2 major reasons. The first reason is that we want to do large-scale species discovery and taxonomy. This is what museums, I think, should be focusing on. After all, a museum is the last place on the planet where taxonomy is being done. So we need to do much more of this. Who else is going to do it? And then, of course, we're also getting specimens that can be used for follow-up research. Among others, we can look at species interactions and find out what these species are actually doing. So one of the oldest questions in biology, and is completely unanswered, is how many species? And it's unanswered, at least for insects, because dark taxa have been ignored. Why have they been ignored? Because with the traditional techniques, it is too slow to work through tens of thousands of specimens. It's not because people don't care, but the techniques weren't there. So what we're trying to do is develop the techniques for exactly these kind of taxa. So you could imagine like a future natural history museum where you never have this entering the building. There's going to be another building where everything is digitized, barcoded, and the specimens are then pre-sorted to approximate species level based on the barcodes before getting to the main building in a pre-sorted state of affairs. Then the real scientific work starts, which is making sure that the barcodes didn't screw up. And the second part, describing the species or identifying the species so that you know what you have in your jar. The workstation for taxonomists could potentially look like this. You have like all this automated imaging being done, and there's different views of each specimen. So you can then train convolutional neural networks to recognize what is dorsal, what is lateral, what is ventral. Then you can make it learn what is a male, what is a female. So as a taxonomist, you could say, show me the male's lateral view of what looks like one species based on the barcodes. And you get then a screen like this. At the same time, you will have information of what barcodes you have and what species is closely related. Because if you try to work with 1,000 species, it doesn't really help you whether there is that you know it's 1,000 species. You also need to know which species do I have to compare to which other species. So you need to have proximity information. And this proximity information can be approximated with DNA barcodes, and that you can get from a diagram like this. Do scientists still have to do this? Can we just describe based on barcodes only? I think I don't have to say too much about it. This was a PhD thesis of Emily Hartop. She had 340 barcode clusters based on all the forward sequencing that we had done. And you see here, there is like some clusters that are incredibly complex. And it turns out that when you take a closer look at them, that these clusters consist of 20 morphospecies. And that happens when you sequence a large number of barcodes for certain clades. But then these barcode gaps that are never really very strong between species start to disappear entirely. Then everything becomes one big morass. And so we do need to have a follow-up study using morphology, but this follow-up study does not have to include all specimens, because if you had to get morphological data for all specimens, we wouldn't gain nothing by this whole process. But what we show in this paper is that you can actually look at only 5% of the specimens and then indirectly check the clusters, the barcode clusters for all motus that are in your study. So it's like acquiring molecular data and image data for everything, then pinpointing to those clusters that are most likely to be wrong. And there are certain ways of doing that. It's described in the paper. And then you just fix the problems with the barcodes clusters that are wrong by looking at a relatively small number of specimens. And that can be done based on morphology or nuclear markers. Doesn't really quite matter. So you have millions of specimens processed. So how do you manage that? That is a giant problem. There was a giant problem in the Singapore lab, but we recently bought this instrument. It's not up yet, so I hope it holds what we expect of it. This is basically a giant box that has space for 200,000 vials. And it works based on compressed air. So imagine you have like one specimen in each of these vials. The compressed air is used to suck up the vials. And as the vials go on the top, there's a little QR code at the bottom of the vial. It's a QR code is being sucked in, or is read in, and then the vial is being placed in this giant carousel. Now, that's only one thing. That's like storing things. That's not the really important part. The important part is retrieving samples. Based on the haplotype information, I know which vials are likely belonging to the same species. So I can give the set of QR codes for the specimens belonging to the same species and retrieve now the species. So it goes in a random order, comes out in approximately species order. Yeah, and if you're lazy, you can also order an individual vial directly to your desk. So it becomes a sorting instrument, right? So the 200,000 vials go in with one specimen each. Initially, we don't know what the species are. We do the barcoding. We know what the species are. We then have them being exported in sets of species. And then we can actually go and store them the same way we used to store them in alcohol collections by putting them into jars. Yes, so this is what we want to do at the center. And I only arrived in August, so we are very far from having really achieved all this. So we want to barcode all specimens, we digitize all specimens, then we get preliminary species boundaries, and we can do the first training of CNNs. The next step is we collect additional data, but only for critical specimens. That can be morphological data, can be nuclear data, in order to resolve problems with DNA barcodes, barcode clusters. Then we revise the species boundaries where the DNA barcodes were incorrect. Then we have to retrain the CNNs for those barcode clusters. And then we do the next thing, which is somewhat controversial, but I think very essential for dark taxa. Then we describe everything. Because now we have actually information from images, we have information from barcodes, and most descriptions, existing descriptions for dark taxa, are inadequate. So we should be redescribing everything. Then you will ask, oh, you're producing a lot of synonyms. No, whether you give a new name, that's the last step, right? So it's not so much, we tend to think about we describe new species, but actually if the descriptions are bad, we describe everything. And then we assign new names for those taxa that are new species to science. Which ones are those? Now that depends very much on, answering that question depends very much how easy it is to answer that question depends on where you are. So if you are in Uganda and you work on phorids, you saw that there's 650 species in just those 8 weeks of samples, only 466 species described. You can go ahead and describe them because the probability of creating a synonym is very, very small. You can't do the same thing for the Swedish phorids. So that's why it's so critical that we are starting to do much more sequencing of types, because then we can identify a lot more of these species immediately. And then it's going to be much easier to also identify which species should not only be described, but also need a name. This will hopefully lead to an automated integrated species descriptions. Because we have images for every single specimen, there's a lot of information there. So even if you get it wrong doing a description, it's not detailed enough. There's a lot of information for fixing the problem later on if a new species is discovered that seems to be very similar to the one that was already described. Now, we also want to know what these species are doing. So for that reason, we want to have specimens. Specimens allow us to do all kinds of follow-up research. We can do microbiome work. We can do all kinds of things. Among others, we could look at diet. So here's another example from Singapore. This was an undergraduate project. This is like an honors project. So you can do this at a large scale because it's not that difficult to do. So this, we barcoded 4,000 specimens of mosquitoes. We found about 120 species, of which about a third could be identified based on blast. Females for about 10 species had visible blood meals. And then, of course, you can meta-barcode the gut content of the females and try to find out what they're actually feeding on. So here's one of those examples. This is a super common species. As you can tell, that's almost like more than a quarter of all the specimens that we had. Found only in 2 different places. So very abundant in some habitats, but not abundant in other habitats. If you meta-barcode the gut content, you find that it is feeding on all kinds of things. So it's feeding on mammals, it's feeding on birds, and it even is feeding on frogs. So that's the kind of species you don't want to see, because this has a very high potential for transferring viruses from one host to another host. And it would be a species to be very concerned about because it's an Aedes species, and a lot of Aedes species are dengue and chikungunya competent. Fortunately, this is quite far away from human dwellings, so that the probability of humans being bitten by that species is quite low. You also find like curious little things like this mosquito, very beautiful. We had quite a large number of specimens, and the specimens from here had blood meals inside, and yeah, they're feeding on mud skippers. So here you have a mosquito feeding on fish. And initially we were like slightly suspicious of it and thought, oh, lab contamination, but there is actually a paper describing this. And then when we looked at Flickr, we sure enough found some mud skipper pictures where people had shown that there was mosquitoes sitting on mud skippers. Mosquitoes do a lot of things. So this kind of work has also revealed recently that they're feeding on earthworms. So mosquitoes are much more Catholic in their taste, in their what they like to feed than most people think. Yes, and we have like a large number of observations for different species. We could get a lot more by sequencing not just the females that have blood meals inside, visible blood meals, because the DNA for the last blood meal will stick around much longer, and you would find a large number of additional observations for some of these species if you were to go large scale. But again, this was an honest project. So eventually what we want for all environments is this kind of thing. We want species interaction networks. We built one in Singapore based on 3,500 interactions for 1,800 species. Most of them were actually citizen science observations. I would not use this for any kind of ecological work because it's super biased, because you have lots of observations for certain taxa and next to no information could potentially have been collected for other taxa. So you have like the macaque having a huge number of observations because a lot of people report on it. But this is the kind of tool that, at least as an outreach tool, is incredibly powerful, because you can show this thing is feeding on that, it nesting in here, and then wherever it is nesting, there's a bee that is responsible for the pollination of that particular plant. So it's a very good outreach tool for understanding that nature is connected, and it's oftentimes connected in just a few nodes apart from each other. So ultimately, we want to get the rich pool divide for biodiversity data to be addressed. And I think the best way to do this is to have largely decentralized biodiversity discovery. What can be decentralized based on the techniques that I described to you is the manual version of species discovery with MinINE barcoding and imaging. because those microscopes and the barcoding are not costing the world. Once we have like trained the convolutional neural network, we can obtain more information downstream using imaging. The facilities that I showed you are expensive because they're like designed for an industrialized nation where the manpower cost is very high. But a lot of these things that we are doing one way here can be made cheaper or can be done cheaper in other places where manpower cost is low. So decentralized should be the labs. Decentralized should be also the taxonomic work. Centralized, I think, should be training courses and data analysis and data storage facilities, so that there's one place where everybody can go and get all the information that is available. And I think we could get very large numbers of species descriptions and image-based biomonitoring techniques rather rapidly if we really were to have a few of these centers running continuously after optimizing some of the techniques that I described to you today. For another year or two, I think we need that optimization phase to get the diversity scanner to be even faster and more accurate. So my overall conclusions are society basically expects us to monitor biodiversity and not just birds. And we have done a really, really bad job at that. Biologists have actually been failing big time here. The major challenge in this context is really our dark taxa, because of the high abundance in the species diversity, which could not be tackled with traditional techniques. Fortunately, I think automation, machine learning, and frugal science can help us change this. We can actually tackle dark taxa. There will be lots of obstacles on the way. Do we have to describe 200,000 species? I'm not so sure. How do we get 200,000 names? That I know is not easy. So there's a whole lot of like things that are coming our way that require a lot more thought. But I think this is possible to at least limit it to 100,000 species. Specimen-based approach, and I think are essential for any kind of habitat in order to not only know what species are there, what the abundances are, but also to figure out what these species are doing. That doesn't mean you don't do meta-barcoding, because the number of specimens you're getting with Malay strap samples is overwhelming. No technique will ever be able to do this specimen by specimen. So it's a matter of like finding the right kind of complementarity for the techniques. I personally would think you start by doing specimen-based work to know what's there. And then the future biomonitoring can then increasingly move into meta-barcoding of whole samples. So ultimately, what I would like to see is something like this, where biodiversity samples are like book samples. You know, you have like a scanner. The specimens come through and it looks at the specimens, images them, is able to identify a lot of them based on the existing convolutional neural networks, after doing all kinds of complicated manipulations to the images. And at the end, you have something like OCR, where you pull the information that is available for the different species, and you can start making sense of whatever change you may have been observing by sequencing samples from the same place over different periods of time. All right, so this is the end of my talk. My acknowledgement slides should be like 5 slides. I make them super short here. This is really the work of numerous people in the Singapore lab, in the Berlin, and also in Karlsruhe, because all the engineering work is being done by the Karlsruhe Institute of Technology, where we are funding multiple PhD projects, where PhD students with an engineering background work on the different robotic devices that are needed in order to make this project a success. Thank you.
Excellent. Thank you very much. Any questions from the audience? Yeah, come on, John.
It's got small things.
Yeah, so it's basically not destructive. Most of the damage is done in the Malay strap. So I'm a dipterist. So I'm basically the Cecidomyias, when they arrive in the lab, they're already heavily damaged. If we do the DNA extraction, which consists of putting the specimen into what is a buffer, heating it up to 65 degrees for 15 minutes, and then 98 degrees for 2 minutes, the specimen does not change very much during that process. That's all we need to do in order to get our DNA. So for that reason, no, the damage is basically not noticeable. I've had taxonomists telling me, are you sure you sequenced that specimen? That doesn't mean that all the specimens are in good shape, but I think it's mostly the damage that is happening before the specimens come into the lab. If something is very fragile, and a lot of the midges are very fragile, then the movement of midges in Malay strap samples is the bigger problem, much bigger problem than the DNA extraction.
I've had the experience of things that have been perfect for Malay straps, going to Paul Eder, coming back mangled.
Yeah, so that's why I don't send my stuff to Paul Eder.
So if I give you some small hyperlocks, will you do some samples so I can see how they come out?
Yeah, sure. I mean, but you guys are establishing the same techniques here, so could easily be done. So the techniques that are being used in Canada are different, although they also now do this DNA leaching. I call it DNA leaching with hotshot. They tended to do, in the past at least, proper DNA extraction. So that includes a proteinase K, and then leaving it in the right temperature overnight or even longer. So it's a much more destructive technique than what we are doing. We're just like letting enough DNA come out that we can do a barcode. We are not even digesting the tissues inside the specimen. And I think the biggest challenge for this process is not really the DNA work, it's handling the specimens. And I think a lot of damage is also created by handling specimens improperly with forceps. So we have different techniques that we use. I mean, a forceps should not be used on insects. Unless it's an expert, right? But if you have volunteers and they're using forceps, the probability of damage is very, very high. Because you also need to have a sense for, this is a hard insect, this is a very soft one, and you need to know how to adjust that. So that's why we are relying on pipetting and a few other tools or tricks for moving specimens that are, if at all possible, not use forceps.
Inez at the back, and then I'll read.
So obviously one limitation is the size of the specimen.
Yeah.
So are you looking into doing the same for sizes, or are you defining anything under 3 millimeters?
No, we're not defining them as dark taxa, but that's actually where the dark taxa are, below 3 millimeter. That's why we started there. So we're going to like define 4 different diversity scanners for different size classes. That will require that the whole sample is sorted by size class, for which there are sieving machines or sieving instruments. You have to be very careful during the sieving process. This is another process where damage can occur. But then we will have 4 different size fractions. The first 2 size fractions can be handled with pipettes. Below 3 millimeter, one type of tip. Between 3 and 6 millimeter, using another type of tip in a different kind of suction mechanism, because you have to have the right suction, the right volume in order to really get the specimen into the pipette tip. Above that, it's going to be diamond pickers. And yes, this is part of one of the PhD projects at Karlsruhe for developing this kind of movement tool. Purely from the point of view of labor, you want to start with below 3 millimeter because the number of specimens that fall into that size class is much higher than the number of specimens that go between 3 to 6 millimeter. And then the larger size classes you could actually handle manually. So that's why this is not a high priority for us to deal with large insects, because large insects make up a very small proportion of your sample.
Alfred.
Yeah, thanks, Rudolf. Yeah, very impressive. But so for the purists, so in a way, you're just targeting one quite specific problem, which is these large clades, things that are known. And so, and when you get this DNA, is there not a problem that we actually use that for many in the future? We use this for many other purposes. So, for instance, we need genetic information. I wonder if that could be somehow built in that vision, which is now very much focused on how many species are there.
Yeah. So it's essentially making sure the DNA doesn't degrade, right? Or having high quality DNA. I mean, yeah. So I had the same discussion with Mara. I mean, it was in Sanger 2 days ago. So they're developing these tools for doing a digestion and doing a proper DNA extraction. That is also very rapid. So we'll definitely be interested in seeing whether that can be upscaled. But the one thing that is good about hotshot is that you're not dissecting the tissue. So we have done some series of experiments where you take a specimen that has gone through DNA leaching, and that's why I call it DNA leaching, and then afterwards been extracted using proper DNA extraction. And we're getting a slight decay in the fragment length, but we're getting a noticeable decay in the amount of DNA you're getting. And for that reason, it doesn't necessarily mean you have to do one or the other. I suspect for the really small specimens, especially for cecidomyids and those kind of things, you probably have to do a proper DNA extraction right at the beginning if you want any kind of good DNA out of it. But for a lot of specimens that are a bit larger and where the tissue is better, better protected, you can do DNA leaching first and DNA extraction next. And our approach has been to then do the more in-depth work only on haplotype level. So you start with tons of specimens, you reduce the problem down to haplotype level or species level, depending on what you are interested in. And then you go in with other techniques that are more in-depth techniques at the haplotype or species level. And that is feasible with hotshot for at least those taxa that are sclerotized.
Thank you, Andy.
So machine learning with your convolution neural networks tends to work very well with fairly two-dimensional specimens. With your automated imaging, how do you overcome having a standard sort of angle of imaging?
Yeah, so I was very surprised that it was that successful based on images of specimens that were falling in all kinds of different directions, but it worked. So to me, black magic, but you know, that's why we have the Karlsruhe guys, because they're experts in black magic. But overall, I think it will become much better if we do imaging from all the sites, because then the teaching or the training of the convolutional neural networks can be done based on standardized views. And that would be the goal. I'm not so sure how scalable that imaging from all sites will be, because it's much slower than picking up a specimen and just moving it into a microplate. So I think we have to think very hard about what we do at what point in time. But if you have like taxonomists working on forests in Berlin, for example, we could decide, okay, for forests, we do the full imaging. For other taxa, we don't. Now, that doesn't mean we can't go back and do the full imaging. So it could be based on projects. So it's okay, we need full imaging now. Let's get the taxa back out and run them through the machine.
Just a quick question, sir. When you identify and describe new species through this, I guess, gold standard, how does that fit into this system?
Yeah, so so I mean, there was a discussion also in Shanghai, as you can imagine. So they're running Malaise traps only for 1 day in order to get higher quality DNA out of the specimens. So based on what I described here, these are like 2-week samples. If you look at the quality of the DNA in 2-week samples, it's already so degraded that long-range sequencing doesn't really work. So then comes hotshot, and it degrades it a little bit worse, makes it a bit worse, but it's still suitable for Illumina sequencing. So if you really want to have de novo sequencing of insect genomes, it almost always requires you to have long-range sequencing. Then this technique will at least tell you where you have to go to find that species. And then you can do what you would do, what Mara is doing, like 1 day Malaise trapping. And you can target then exactly the time period when you suspect that in this particular place the species will show up. And then, of course, if the convolutional neural networks go to species, which I am kind of doubtful, you could then use images alone to identify the specimen that you were looking for in order to do the de novo genome sequencing. But at the bare minimum, it'll tell you you have 20 specimens in that genus you're looking for. I think to genus level, I'm pretty confident that the convolutional neural networks will go. Then you extract those 20 specimens, and then you do a quick barcode, and then you know which one it is where you want to go in depth. So I can see this being useful for that purpose, but this is not the main purpose here. And I think doing genomes for 200,000 species of forests in Africa, I think is also it's a different question.
Yeah, exactly.
I mean, for larger specimens, it's also so that the hotshot leaching that we do only involves the head of the insect. So we just put the insect in and the head is in the hotshot, the rest isn't in the hotshot. So you can get a ton of perfectly normal DNA out of it afterwards.
As you increasingly train your AI system, at what point do you think that so you're already going from 2D to 3D, I mean, go from there to maybe hyperspectral and CT scans, at what point do you not need the opposite question? You don't need any sequence at all. And basically the imaging, whatever way it is, work it out. And do you have any feeling from the types of studies that you've done about the level of image data that you need to basically get the species level?
I have very poor sense for that because I'm not an expert in that area. So the only place where we have done something in that direction was tick identification in Singapore. So we had a few thousand specimens. We barcoded them all. We then did convolutional neural network training to distinguish the different stages between larvae, nymphs, and adults, then between males and females, and identifications are done on females. And then in the end, we had like females for 16 species. And of those, I would say 13 could be identified with a probability well above 90%. And then there was 2 that were terrible, like 40% success rate. And there's one that had like 70% success rate, which is, I think, what I would expect. You know, so you will eventually have like a proportion of species where you cannot use the convolutional neural networks. And then there's other species where it is no problem. And I don't think anybody has a clue what proportion will be in what class. But I'm also usually say that it is not a matter of the networks, it's a matter of the imaging that you give them, right? So the alternative is to have a diversity scanner that knows where the rear end of a male is, and then takes another image of the rear end of the male, or maybe even a confocal image that has stacking information. And then you can train the network based on imaging information that also humans would need in order to identify that species. But how scalable that is yet another issue. So, I mean, I have plenty of things to do with. So, I mean, I have ideas about these things, but I don't think I will ever get to them.
I got to know your work from a mutual friend who told me about the system. And it struck me that he told that the machine, in the end, picked up different diagnostic features with respect to those used by taxonomists. Is that true? And so,
yeah, so
can also taxonomists be trained to look for some other characters?
Yeah, so I think that's why the heat maps are interesting. Whether they're informative for taxonomists, I'm not so sure, because it's oftentimes shape information. But you know, the one thing that makes me hopeful that these things will work fairly well is that when a taxonomist goes in the field and he sees something running around, then he can oftentimes say what it is, and that comes from somewhere. And it's oftentimes shape characters, sometimes also how they move, and of course a probability statement of is it likely to be here, and that leads to a species identification. And what the CNN does to some extent is more like that. So it's more looking at shape characters, which we avoid in taxonomic work because it's hard to describe. It's hard to describe also the variance in the species. So if you can find something that is qualitative, you're much happier. So in a way, we have been looking for qualitative characters, but that doesn't mean there is no quantitative characters. And so my hope will be that the quantitative shape type characters will be picked up by the algorithms. And yes, the heat maps give you some information of what other thing you may want to look at, because the machine is using something different from what you would be using.
Brilliant. Any other questions before we wrap up? Chris.
I guess kind of a political one, actually. This is very compelling and impressive, and I think you make a really great case for how this can scale up the enterprise of taxonomy. But this is a system which lends itself really well to this kind of automation. There's a huge part of Earth's biodiversity which is just intrinsically going to be a lot more labor intensive to describe. Parasites, species in low abundance, deep sea settings, stegopitic fauna and cresting fauna. I could go on. You're making a pitch, it sounds like, or thinking about trying to set up centralized facilities to do this that, gives us accountable outcomes very rapidly. Is there any part of you that worries that this might, become a hammer that hits only one nail, attracting from attention to other nails that don't have hammers like this?
No, basically the lack of hammers means that we are not doing very well in taxonomy, right? So if we have like one hammer that is actually doing dealing with one of the nails that is like making holes into our shoes, then I think that is better than not having any hammers. So to me, this is targeting things with exoskeletons very clearly. Deep sea, yes, I can do deep sea corpopods. That's not an issue. But this is really for arthropods. Because that's where I can get DNA without destroying the specimens using a leaching technique. Now we all have to kind of select the taxa or the work that we do. And you can make that argument about everything, right? So it's always like by being good in one area, you neglect some other area. At my age, there's not many years left. So you just like pick a minor little problem like the arthropods, and you're perfectly happy to develop that hammer and worry much less about the other hammers. I mean, of course, it will distract from others, but I mean, that's the case in science all the time. It's always competition for funding. But if you can go further with the tool in one area, the fact that the other area may not be moving as fast is actually the right thing in the overall picture of scientific development or knowledge development.
Hopefully this is a rising tide that will rise all boats.
I mean, there are like similar things that can be done with other taxa, I'm sure. It's just somebody else has to think about them.
Excellent. Right, if there are no more questions, we can wrap up now. I think another round